Next topic, molecular clocks. The Holy Grail. Fossil evidence is sparse and imprecise or non-existent. Predict divergence times by comparing molecular data. Given a phylogenetic tree, branch length RT, a time estimate for one or more node, 110 MIA. Can we date other nodes in the tree? Yes, if the rate of molecular change is constant across all members. Rate consistency given like this. Protein variability. Protein structures and functions differ. Proportional of neutral sites differ. Protein structures and functions differ. Proportion of neutral sites differ. Rate consistency does not hold across different protein types. Protein structures and functions differ. Proportion of neutral sites differ. Rate consistency does not hold across different protein types. However, each protein does appear to have a characteristic rate of evolution. Evidence of rate consistency in hemoglobin. Molecular divergence time. Black dot represents fossil divergence time. Baboon, cow, chicken, alligator, frog, carp, shark, large carnivorous marsupial. The molecular clock hypothesis. Amount of genetic difference between sequences is a function of time since separation. Rate of molecular change is constant enough to predict times of divergence. The molecular clock hypothesis. Amount of genetic differences between sequences is a function of time since separation. Rate of molecular change is constant enough to predict types of divergence. Outline methods for estimating time under a molecular clock. Estimating genetic distance, determining and using calibration points, sources of error, rate heterogeneity. Reasons for variation, how it's taken into account when estimating time. Reliability of the estimates, estimating gene duplication times. Measuring evolutionary time with a molecular clock. Estimate genetic distance, denumber amino acid replacement, use paleontological data to determine date of common ancestor, time since divergence, then estimate calibration rate, number of genetic changes expected per unit, calculate time of divergence for novel sequences. First is measuring evolutionary time with a molecular clock. Estimate genetic distance. Use paleontological data to determine date of common ancestor. Estimate calibration rate, number of genetic changes expected per unit time. Calculate time of divergence for novel sequences. Estimating genetic differences. If all NT equally likely, observed difference would plateau at 0.75. Simply counting differences underestimates distances, fails to count for multiple hits. Sequence difference, expected difference, observed difference, time. Estimating genetic distance with a substitution model accounts for relative frequency of different types of substitutions, allows variation in substitution rates between sites given learned parameter values, nucleotide sequence frequencies, transition or transversion bias, alpha parameter of gamma distribution, can infer branch length from differences. Distances from gamma distributed rates. Distances from gamma distributed rates. Rate variation among sites. Fast variable sites, third codon positions, codons on surface of globular protein, slow or invariant sites, tryptophan 1 codon structurally required, first or second codon position when disulfide bond needed. Alpha parameter of gamma distribution describes degree of variation of rates across positions. Modeling rate variation changes, branch length, sequence differences curve. Gamma corrected distances. High rate sites saturate quickly. Sequence difference rises much more slowly as the low rate sites gradually accumulate differences. Then Felsenstein inferring phylogenetics. The slopy clock. Ticks are stochastic, not deterministic. Mutations happen randomly according to a Poisson distribution. Many divergence times can result in the same number of mutations. Actually, over dispersed Poisson correlations due to structural constraints. Poisson variance, assuming a perfect molecular clock. If mutation every MY, Poisson variation 95% lineage, 15 MYA old have a 8 to 22 substitutions. 8 substitutions also could be 5 MYA. Need for calibrations. Change rate into time. Can explain any observed branch length. Fast rate short time, slow rate long time. Suppose 60 changes along a branch could be 2 into 8 or 8 into 2. No way to distinguish. If told time 8, then rate is equal to 2. Assume rate is equal to 2 along all branches, can differ all times. Estimating calibration rate. Calculate separate rate for each data set, species or genes using known date of divergence from fossil biogeography. One calibration point, rate is equal to d by 2t. More than one calibration point, use regression. Use generative model that constrains time estimates. More later. Calibration complexities. Cannot date fossils perfectly. Fossils usually not direct ancestors. Branched off tree before or after splitting event. Impossible to pinpoint the age of first common ancestor of a group of living species. Then linear regression, fix intercept at 0, 0, fit line between divergence estimates and calibration times, calculate regression and prediction confidence limits. Molecular dating sources of error. Both x and y values only estimates. Substitution model could be incorrect. Tree could be incorrect. Errors in orthology assignment.